Hello, everyone, and welcome to this webinar in which we will be talking about clustering analysis to study transmission of tuberculosis. So starting from the beginning, clustering is based on the simple concept, which is this patient who is infected with tuberculosis transmit the disease to another patient. Of course, that means that he or she transmits the bacteria that causes the disease to this other patient. And because of this, of course, these two patients harbor the same strain. And because MTB is clonal and presents no recombination and a very slow evolution and has a very low genetic variability, we expect few genetic differences between strains that have been recently transmitted. So we would expect that these two red strains really are genetically very similar. And just one thing to bear in mind, which is note that recently here is involved. And we will come back to this concept once and again, so you understand that clustering is talking about recent transmission. So what are we talking about when we talk about clustering analysis? We perform clustering analysis when we want to know whether tuberculosis is transmitting in our population. So we want to know whether there is ongoing transmission of tuberculosis in our study population, and we want to detect outbreaks of tuberculosis. So the way we would do clustering is we have a sample of the population that we want to study, and the patients of tuberculosis will carry given strains of the bacteria. We perform strain typing, and then now we know which strains each patient is infected with. And because these two patients share the same strain and these two patients share the same strain, we can say that these are in the same transmission cluster, these are in the same transmission cluster, and this patient is unclustered. Now, what does it mean to be within a cluster or to be unclustered? When patients are within the same transmission cluster, what that means is that those cases are linked by recent transmission. So they belong to the same transmission chain. They belong to the same outbreak. This is already a very useful information, but bear in mind, and it's important for you to always remember this, that this doesn't mean that transmission is happening between these patients in particular. And the reason is that we cannot rule out the possibility that there is another patient or other patients that we did not sample, so we don't know them and we did not sequence this strain, but actually these patients transmitted tuberculosis to these others. So again, clustering means yes, they are in the same transmission chain and they form part of the same outbreak, but not necessarily transmission happened from one patient to the other. So this is a very important point here to notice, which is clustering does not tell us how transmission occurred. So cannot tell us what the direction of transmission is or who infected whom, cannot be used to delineate the transmission chain and know that information. So let's see more into detail what clustering means and what it does not mean, and what it means also to be not within a cluster. Let's consider the following scenario. So we have two countries, country and region, or region A and country region B, and we want to study tuberculosis transmission in country B, right? So we recruit the set of patients, or we perform our sampling. And of course, one thing that is already important to note is that you can never sample all the entire population of tuberculosis by many reasons. It's just not feasible. So your study population or your data set will be always a subset of all the possible cases of tuberculosis that are circulating in the population. And this is important, and you will see why it is important in the next. We perform strain typing, so now we know that these patients are infected with the red strain, and these patients are infected with the magenta, the blue, and the black strain. So according to what we saw in the previous slides, these patients form part of the same cluster, so they are linked by recent transmission, or they form part of the same outbreak. Again, remember, we cannot say who infected whom, or how transmission occurred, or even if it occurred between these patients, because maybe there is another patient who transmitted this strain to these patients. Now, we know that there is a recent transmission or ongoing transmission of the red strain. So what is happening with the magenta, the blue, and the black strain? We cannot know which strains are infecting these patients here, because of course we did not sample them, but let's imagine we could know. So given this situation, now let's try to know, and let's try to explain why these 3 patients were unclustered in our analysis. So the magenta strain was not clustered because there are no other magenta strains circulating in our population. However, the magenta strain is very common in country A. In fact, there is ongoing transmission of the magenta strain. But this was a case of an importation. So this patient who was probably living in country A moved to country B where the magenta strain is not circulating. He or she developed active TB, and then because there are no other magenta strains, it is unclustered. But this is, it is important for you to realize then that clustering is not telling us whether magenta strain is transmitting more or less. Actually, there is recent transmission of the magenta strain in country A. And the reason it is unclustered in this country is not, again, because it's transmitted better or worse. It's because it's an importation, and this magenta strain is not circulating in country B. In case of the blue strain, what happened is something that it's also very important to bear in mind, because clustering is really sensitive to this kind of bias, which is the sampling bias. So as you can see, the blue strain is circulating in our population actually, and there is recent transmission because we find different patients that harbor the same strain. So the reason in this case is unclustered has nothing to do whether the blue strain is transmitting more or less. In fact, does not even has to do with whether there is recent or not recent transmission. Actually, there is recent transmission, but the problem is we did not sample these other patients that would be in cluster with this patient. So always bear in mind that the sampling of your population is very important, and if you have lower samplings, your clustering analysis can be highly affected by this bias. Finally, another reason why a case could be unclustered, and well, at least another reason you will find in the literature why a patient could be with unclustered, would be a case of reactivation of a latently infected patient who was infected many years ago with a strain that was circulating at that moment in the population, but that strain, the black strain, is not circulating anymore in our population. So when this patient develops active TB, that strain is not in cluster because, again, there are no other black strains running in that population anymore. So that could be a case of a reactivation of an old. I would like to dedicate a couple of slides to talk about a concept that I find that sometimes is a little bit misleading. I might have explained this concept partially in previous slides, but I find it might be interesting and it might be important to clarify this. Clustering is very useful, again, to investigate outbreaks and to investigate transmission of tuberculosis. And with that, we can also investigate traits that might be or might not be related to transmission, like, for example, drug resistance. Is it drug resistance being transmitted or not, right? And it is in that sense useful. Now, but when we say that we are studying transmission using clustering, are we detecting transmission as opposed to what? Are we detecting transmission as opposed to non-transmission? Because conceptually speaking, tuberculosis is always transmitted. There are no natural reservoirs um, for tuberculosis, so you don't get tuberculosis from wastewaters. You don't get tuberculosis from a mosquito bite, as it happens in malaria. You get tuberculosis because another patient infected you with tuberculosis, right? So always a case of tuberculosis is coming from another case of tuberculosis. So conceptually speaking, all tuberculosis cases form part of the same transmission chain that has been running through the history, right? So then if we cannot, if then they are all from the same transmission chain, why is clustering useful? Well, the thing is, the bacteria that causes tuberculosis, of course, has been evolving through the history and through the years, and it evolves, still evolving through the years, and as it evolves, it accumulates mutations. So when enough time happens, um, it accumulates so many mutations that at the end of the day, they are different, genetically speaking, they are different bacteria. So the let's call the original bacteria was one, like this red bag here, but now it accumulated so many mutations that now it's this other bacteria here that is genetically completely different. So we can really differentiate them, right? So clustering is useful to talk about recent transmission and to, again, as you saw in the previous slides, to differentiate it from imported cases, for example, reactivation, or just differentiate whether one strain is being transmitted in our population or not. The reason is, if two strains are genetically very, very similar, that means they did not have time to evolve enough mutations so we can differentiate them. So we can say that they are the same genotype, and they are the same genotype again because very little time happened for them to evolve and to differentiate between them. So that's the reason when we see two patients sharing the same genotype, we say, well, this is due to recent transmission, because the strains did not have time enough to evolve many mutations so we can consider they are different genotypes. So, as I just said, clustering can be very useful to study some particular biological traits, like, for example, drug resistance, which we might be very interested in, right? So I will explain now how clustering can be used to differentiate or to study de novo versus transmitted resistance. So I will start by explaining these concepts. So imagine this patient, John Doe, has a susceptible strain, and um, he receives antibiotic treatment. And because antibiotic treatment is a strong selective pressure, drug-resistant bacteria evolve within this patient, and this strain that was susceptible at the beginning now is drug-resistant. So this is a case of de novo evolution of drug resistance, which is also called, you will see that in the literature, particularly epidemiological papers, it's also called acquired resistance. There is another possibility, which is John Doe has this blue strain, which is already drug resistant. And he transmits to Jane Doe, and now she got drug-resistant tuberculosis, but it's not because of the drug treatment that Jane received, it's because she was already transmitted with a strain that was already resistant. So this would be transmitted resistance, or the technical word for this is called primary resistance. So how can we use clustering to study these two types of resistance? So the novel evolution within a patient due to drug treatment versus transmission of strains that are already resistant. So let's consider the following scenario. We have these 5 cases, and all of them are rifampicin resistant cases. We have already typed the Mycobacterium tuberculosis strains that are infecting them. So we know these, and that means they are all within the same transmission cluster. So because they share the same strain, and we can say that they are linked by recent transmission, and there is ongoing transmission happening of the red strain, the most likely scenario is that what happens here is that the drug-resistant strain was transmitted. So these are cases, or we could say that there is transmission of drug-resistant strains happening here. In these cases, because we did not find other patients clustering with these, so we do not detect in our data sets ongoing transmission of the yellow strain and the blue strain, we would say that most likely these patients acquire resistance de novo. But again, it is super important that you bear in mind what the limitations of clustering are. We will always talk about likely scenarios. So we know we can use clustering as a proxy. Yes, it's useful, but it's not the ground truth. Consider that there are sampling bias, there are imported cases, and this yellow strain, for example, in this patient, maybe this patient came from a country where multi-drug resistance has a very high prevalence, and maybe this was also a case of transmitted resistance, but now this patient moved to another country where this yellow strain is not circulating. So we would say it's a case of de novo drug resistance, but that would be a mistake because actually it was a case of transmitted resistance. Not saying that we cannot use clustering. Clustering can be used as a proxy, as we are representing in this slide, but always bear in mind the limitations. Now let's consider another possibility to make this a little bit more complex, which is the same scenario, but remember we are using whole genome sequencing, and with whole genome sequencing we have increased resolution, so we can look at specific mutations, and we see which specific mutations are conferring drug resistance to these strains. And we see that within this red transmission cluster, strains have different mutations conferring rifampicin resistance. If they were cases of transmitted resistance, necessarily they would share the same drug-resistant mutations, but this is not the case. So the most likely scenario in this case is that these are cases of de novo resistance also, despite being in cluster. So most likely these patients were infected with the same red strain that was susceptible, and then as a consequence of drug treatment, different drug-resistant mutations evolved within the different patients. Now, so far we have been talking about strains in code color, so the blue strain, the red strain, the yellow strain, and so on. But when we talk about strains, or when we perform strain typing, the question we are trying to answer is whether two given MTB strains are of the same genotype or not. And of course, with this question comes another question, which is, what does it mean to be from the same genotype? And how we consider genotypes will also depend on the tools that we are using to perform the strain typing, right? So we could be using IS6110 RFLP patterns that have been widely used in molecular epidemiology, as you might know. For example, we would have all of our strains in which we perform the RFLP experiments, and then genotypes. So what our consideration of genotype when using IS6110 RFLPs would be whether two strains share the same RFLP pattern or not. So for example, these two strains share the same RFLP pattern, so they would be considered the same genotype, and then in this case they would be considered to be linked in the same cluster, RFLP cluster, and then ongoing transmission or recent transmission. And the same happens for these two strains here, the ones highlighted in blue, that share also the same RFLP pattern. Of course, the problem by using this method is that we are only interrogating the IS6110. So our definition of genotype is not based on all the information that the genome actually can provide us, but only a small portion that corresponds to this insertion sequence. So as you may know, the resolution of the different tools um, that have been used in molecular epidemiology, or that they are still used in molecular epidemiology, varies. So different tools have different resolution depending on the proportion of the genome that they interrogate. But of course, because of the resolution depends on this proportion of the genome, whole genome sequencing, which is by definition the analysis of the complete genome, is the one that provides the higher resolution because it provides us resolution at the SNP level. So we can detect and we can do clustering analysis based on a resolution of single SNPs. So now, with increased resolution comes also kind of a problem, which is the following. We are able now to analyze difference at the SNP level. So imagine the next scenario. So we have two strains that we want to compare. They are coming from two different patients, and what we do is we perform whole genome sequencing of them, and we compare their genomes, and we see there are 0 SNPs difference between them. And what that means is that their genomes are 100% identical. So they are exactly the same genome. And then there is no discussion. We can find another situation, which is we have, again, two patients. We perform whole genome sequencing of the strains that are infecting these patients, and we found 1,000 SNPs difference between those strains. So maybe you don't know so far, but maybe you will know at some point if you keep doing clustering analysis and analyzing genomes of MTB bacteria. 1,000 SNPs of difference between two strains is a huge difference because, again, as we explained, mycobacterium tuberculosis evolves very slowly, and we don't expect in short times to find high amounts of SNPs of difference. So again, there is no discussion here with such a high distance in SNPs or high difference in SNPs that these are different genotypes. The problem, of course, is where to put a limit. So the problem is how many SNPs do we consider that can define recent transmission? So in short times of evolution that happen when one patient transmits to another patient and that patient develops active TB disease that might happen, several years can happen, how many SNPs at most we expect so we can put a threshold that defines recent transmission? In other words, what is the SNP threshold at which we consider two strains to be different genotypes, or the other way around, to be the same genotype? Because this might be a little bit complicated, and actually it's a complete topic in tuberculosis analysis and, well, clustering analysis of mycobacterium tuberculosis. This is something we will be covering in the next webinar. So again, I hope you enjoyed this webinar talking about clustering analysis, and we'll see you in the next one talking about SNP thresholds.